Okay, just hold on. I should call Pritodip once. He has been removed from the meeting for some reason. হ্যাঁ প্রীতদীপ আবার জয়েন করো। হ্যালো, আরেকবার জয়েন করো। তোমাকে একটা টেকনিক্যাল কারণের জন্য ওরা বের করে দিল রেকর্ডিং এতে, আবার জয়েন করতে বলছে। So today my topic is cell membrane and its derivative in protists. This is a very new topic in this paper. So far, it is a new way of compilation. Nothing is, the information is not new, but such a topic I have not come across such a heading or topic before. This is according to the new LOCF syllabus in this paper. So in this paper, what it does, what it means, that what is cell membrane and what are its derivatives in the cell membrane. So we will first look at the protozoan kingdom, the protists, the unicellular organism, single cell, where all the functions of the cell are performed by individual organelles which are present in the cell, and there is only one cell, and that cell performs all the physiological work. Now, keeping this in mind, we will go through little groups. First, we will try to see what a cell membrane is and what is the structure of a cell membrane. And then it's the same as in your cell biology course which you have studied. And then we will actually go through what happens through different groups of protozoans. What are the different derivatives? So, what do you mean by derivatives? Derivatives means what are associated with the cell membrane, that is what are derived from the cell membranes. For example, in protozoans, the cell membrane along with certain microtubules and underlying cytoskeletal elements together, and some external elements, they form a structure known as the pellicle. So whenever we talk about a pellicle actually therefore is a derivative of the cell membrane, because the pellicle also contains the cell membrane. So we essentially will be studying associated structures with pellicles. जो स्ट्रक्चर पेलिकल के साथ एसोसिएटेड है, हम उसको लेके हम लोग बातचीत करेंगे. So what is the pellicle? The pellicle is सेल के अंदर माइक्रोट्यूब्यूल्स होता है, साइटोस्केलेटल होता है, which gives the cell a particular structure, a shape, and it performs certain functions inside the cytoplasm, which we saw in cell biology. We have read about microtubules and what are the things which are present in the microtubules. We have studied all these things. And now we will study all the associated structures with the cell membrane or the pellicle. It can be a covering, a test. A covering is known as test, T-E-S-T. It is not an examination test, but a covering. Hard covering is known as test. So we will be talking about that. Different types of how in amoeba pseudopodias are formed. Then what is the shape in euglena. Then what are foraminiferance. What are their unique characteristics, the shell, the covering. Then we will also be talking about the different cilia in paramecium. Then what happens in plasmodium and in other parasites like trypanosoma, how are the cell membrane organized. So this is a absolutely very new topic which we are studying. The information is nothing new, but the topic definitely is new. This is for the first time somebody is teaching such a thing. I'm quite sure I may be the first person teaching this, because this is the absolutely new syllabus. It has come out, and so far in my information, this has not been covered in any class. And this is the first note which has been made, and I would request you not to share this note with your other students until or unless it is published first. because it's right now confidential, in a confidential mode. So you people study it, read it, but don't share it with anybody right now. Not at this moment, because then you will fall under an issue known as copyright, and that goes under a very serious legal proceedings. So just be very careful. You study it, write in your examinations, in your projects, whatever you can use it freely, but don't share it with people, because some of these people, they may share it again for monetary gains, and when they share it, this will fall under copyright protection, and this is not good. So once it's published, you can do anything with it, giving the reference from where you have taken and whatever it. But right now, be very careful. Read the note. I've sent it on WhatsApp. I'll publish it perhaps by the end of this semester. It will be published, but you people will get it before it is published. So let us have a look at what is the particular note. Let us talk about this note. So I'm sharing my screen right now. Anisa, bolo question. हाथ उठाया, raised hand. Nisab, any questions? Okay, anyway. So let me enlarge it. Let us have a look at the structure of a cell membrane. So this is the structure of a cell membrane. What you have first is, this is one layer. This layer is the outer layer of the cell membrane. Okay. Then you have this is the lower layer of a cell membrane, and therefore they are known as, it is known as a bilayer. It is known as bilayer. Therefore, it is known as bilayer, and it is a lipid. This is one lipid bilayer. So these little tails which you see over here, this is the
phospholipid.
Exactly. This is the phospholipid. Phospholipid layer. And this phospholipid layer, so this phospholipid layer consists of two parts. This outer part, this is a hydrophilic. What do you mean by hydrophilic? By hydrophilic, it means it loves water. So this outer ball-like structures which you are seeing, they contain the phosphate group and molecules like ethanolamine, serine, inositol, uh, then choline. These molecules, they love water. So the outer layer is hydrophilic. They like water. Therefore, as you know, the cell lives in an environment which is aqueous. The outside is a liquidy layer, and that layer basically likes water, so it's towards the outside. And the inner, these little leg-like structures, you see, they are hydrocarbon chains, and they hate water. They are hydrophobic. So the inner side of this membrane, this is hydrophobic, and outer side is hydrophilic. Now you have several proteins present. So where are the proteins? Let me remove everything. So I'm removing all the junk which I've written over there. So these are the proteins here. This is a protein. This is a protein. So this is a transmembrane channel protein. Transmembrane is that it spans from this is the outside and this is the inside region. Outside region and inside region here. It spans throughout the membrane. This region, center region, it is hydrophobic, and the outside amino acids, they are hydrophilic. These are hydrophobic amino acids. They hate water, and these are outside. They form a channel to allow passage of substances. These are transmembrane channels, and they are also known as integral membrane proteins. This is also a integral membrane protein here. Apart from that, you have proteins which are associated temporarily on one side. This one or this one. These are known as peripheral proteins. Apart from that, on these phospholipids, you have carbohydrate chains attached, and they are known as glycolipids. Even on the proteins, you have these carbohydrate chains attached. These are carbohydrate chains, and they are known as glycoproteins. Protein plus lipid, carbohydrate, protein plus carbohydrate, glycolipid, protein plus lipid plus carbohydrate. What is it? What is a lipid plus carbohydrate? Can you tell me? Can anybody tell me what is lipid plus carbohydrate? Glycolipids. And lipid plus this protein plus carbohydrate, glycoproteins. So you have these things. They can act as signaling molecules also. And in between, you have molecules known as cholesterol. These cholesterol molecules, they account for membrane fluidity. The membrane is like a mosaic. You see your floor, it's a mosaic floor. If you have got a mosaic floor, you will see that on a white or yellow background, you will have lots of black and dark and colored little color spots. You can have lots of color spots. So this is a sea of lipid, which is and inside the sea of lipid, you will find like mosaics, these little proteins are attached, and the nature is fluid like. It is neither solid, neither liquid. Horhare, holthale. So it's a fluid-like structure, and this fluidity is maintained by this cholesterol at different temperatures. If it becomes cold, it still maintains the fluidity. It becomes hot, it still maintains the fluidity. So this is maintained, the fluidity is maintained by cholesterol in between. What it does is it just, when it becomes hot, these all start to move about. So this cholesterol molecule inside the center, it prevents the movement very freely. When it becomes cold, what this cholesterol does, it spaces these molecules to the hydrophobic tails far away from each other, so that they just don't contract and become frozen at lower temperatures. So this is how it works. But for absolute low temperature, it will freeze and it will break. So the fluidity is very important for a cell. So this is the cell biology part of it. We go with the cell biology part. Now let us come back to the our protozoan part. So there is also one more line which I would like to mention here. Lipids, proteins, and carbohydrates on the outside surface form a coat which is extremely important in defense against external chemical attacks, and it is known as glycocalyx. So this outer covering region, which you see over here, this outer region with all these things, they have got a particular signature, and it is known as glycocalyx. And it is known as glycocalyx. This glycocalyx in different organism may be different, and they elicit an immune response say if it's a parasite and it enters your body, it will induce an immune response from your body. This glycocalyx is very important for the host's immune response, and against the external chemical attack for this organism, some chemical, they will prevent the damage to the cell, this glycocalyx. The next important line which I've kept it in bold, I'm highlighting it, is the cytoskeleton elements, or like protein filaments like actin, which is present inside, and the cell membrane in protozoans, they form a body wall known as pellicle. So these unicellular organisms, they have a body wall called pellicle. হ্যাঁ প্রীতদীপ কিছু বলবে?
No, sir.
Okay, okay. So they form a cytoskeletal elements, that is the microtubules which you see, actin, myosin, all these cytoskeletal elements you will find inside the cell cytoplasm. They, together with the cell membrane which we have studied, they form a body wall known as the pellicle. And now we will be discussing about certain derivatives found in the protozoan kingdom. Now let us see what is in euglena or euglenoids. In euglenoids, have a look at this picture. Have a look at this in this picture. If you look at the surface of an euglena under my electron microscope, you will find ridges like this, these lines obliquely. And now if you go to a cross section, if you cut a euglena and under electron microscope, you will find these knob like structures, door knob, दरवाजा का जो घंटी होता है उसको पकड़ के जो घुमाते हो, these door lock like knob like structures, and in between these knob like structures you will find here grooves, and these grooves, if you elongate this, these basically grooves are what you see as these oblique lines. So this is how the pellicle or the cell membrane along with the other associated structures, they look like in euglena. So the movement of these things, they result in a peristaltic euglenoid movement, a type of circular movement like this. It happens, and that is responsible for movements. Now we will look at what happens in kinetoplastids. So the kinetoplastids are like parasites like trypanosoma, which causes sleeping sickness, and leishmania, which causes kalazar. So these what happens there? How they look at this is a trypanosoma. This is very interesting. If you look at the outer surface or the the outer membrane and its derivative surface of these trypanosomes, you will find interesting structures present over there. The first thing which you find is this region, which I am making it in highlighting in yellow. This is an undulating membrane. It is a fold of the pellicle. The pellicle folds into a membrane-like structure known as undulating membrane, and it contains. It has got a flagellum. This is the flagellum which you see, and you can find that the flagellum, this red line, this line which is passing like this. This is the flagellum is attached. So this undulating membrane has got a fold of the pellicle as well as undulating membrane. This is important for the movement. Now, apart from this, what is very important is that pellicle has a thick glycocalyx which has over which has proteins encoded by 1,000 genes. The pellicle has a thick glycocalyx which has proteins. This over is, you can erase this over here. It's a typing error. I will remove it later. So this has proteins encoded by around 1,000 genes, which make up 40% of the genome. So, say when this organism is infecting a person, it enters into the bloodstream. Immediately, what happens? You have cells, an immune system of your body, which will make antibodies against the glycocalyx, which has got over several proteins, unique proteins. They will identify these proteins, our immune system. Okay, this is a trypanosoma, and they will make an antibody against this, and they will destroy this cell. So, for the parasite to establish infection in that person's body, it becomes impossible. So, the parasite has got a mechanism to evade this. What it does is, during infection, it will display a different set of proteins on its surface, on the cell membrane. So even the immune system will try to make antibodies against those particular proteins. Now they have made a particular time. It will take some time, 1 or 2 days time, to make this particular antibody. What is wonderful over here is that by the time antibody is made against these, say, 20 proteins, which was expressed, this organism, it goes to the next stage of infection, and it changes the proteins. So the former proteins against which the antibodies were made, they are gone. You have a new set of proteins being made, and the organism, it escapes the first line of defense. Now the new set of proteins is, then the immune system, what it does, it will try to make proteins, antibodies against this new set of proteins. So by the time the antibodies are produced, another set of proteins are made. So wow, so many antibodies are made, but the organism, it evades the immune system, and it establishes infection inside the host of that person. This is known as these are differential expression of proteins. There are around 1,000 of these proteins made by 1,000 genes. Different of them, maybe 10 of them, 20 of them, at different infection stages, they are being expressed. And these proteins are known as, they are glycoproteins. What are glycoproteins? Proteins plus carbohydrates is glycoproteins. They are known as variant surface glycoproteins, VSGs. This is how the pellicle or the glycocalyx has got a wonderful way. So you can see that the first set of, let's highlight, this VSG is made, variant surface glycoprotein is made, immune response is there, it increases. By the time it does, a second set is made. In the next step, a third set is made. So the parasite titer falls, and again a new set is made, again it increases. So you can see that the immune system is always working, and there is always a conflict between the host and parasite. So all in this glycocalyx, or the cell membrane derivative, all the function by which they escape the immune system. The next one which we study are the Ciliophorans or Paramecium. Their body is covered with cilia. So you see, this is the cell membrane over here. This outer region which you see over here, that is the cell membrane. This is the cell membrane, outer region, the cell membrane. It's a covering. So what the cell membrane does, below the cell membrane, there are these sac-like, this momo-like shape, structures known as alveolus. And there is a space in between known as alveolar cavity. It's a membranous sac. And through this membrane and this, this several they are arranged like sac-like cavities on top of the paramecium body. This is the cross section, and you have several cilia or locomotory organism coming out from the center. This is the system in Cilioforans. The next is we talk about apicomplexa. Malaria, Plasmodium. So that is also fantastic. You have an outer membrane. This is the outer cell membrane. The outer cell we have the outer cell membrane over here. Below that, there are two layers of another underlying membrane, inner and outer membrane. They enclose this a sac-like structure. Let me increase the size and I will see if you can see better. You see the outer layer. This is the outer layer of cell membrane, and underlying it is the alveolar inner two layers of membrane. But you see one layer only here, but there are two layers. And this layer, they are open at one region, this polar region known as apical complex. Therefore, the in Plasmodium or malaria, which causes malaria, there is a space. This is the apical region. Another space is in this laterally in the micropore region. This membrane is not connected, joint. And another site is in this region, is the site of exocytosis. So the inner membrane which you see, it is sort of not continuous, it's broken. This is one region where it is broken. This is another region where it is broken, and this is another region where it is broken. So this is what happens in apicomplexa, or the ones which cause malaria. Now the most important part, where the one which I think will come in your examination, are what happens in amoebas. You know amoebas? So this amoeba protozoans, there are three types basically. The amoebas, which you see, the amoeba proteus, the foraminiferance. Very speciality of this foraminiferance is that their body is covered by a test. And you have actinopodons. They are also another type of amoebans. So the first thing which you see is Lobopodia. So the Lobopodia is found in the normal amoeba proteus. They are projections which are wide, rounded, with the tips which are blunt. So look at this shape over here. It is blunt, mota. This is blunt, rounded. The tip is blunt and rounded, irregular shape. This is a Lobopodia. This is a type of Pseudopodia. Pseudopodia are finger-like projections. Everywhere it is not like finger. It can be needle-like also. Then you have Philopodia. You see filament-like projections coming out. You see in some, for example, you can find it in Penardia mutabilis. You can find in, this is also another amoeba which shows this finger-like projection, the filament-like projections coming out. They're phylopodiums. Now let us look at the test or the covering in some amoebas. There may be an organic matrix with studded siliceous elements. So let us look at one in Arcella. This is Arcella. This is Arcella. In Arcella, you see its body is covered by a test or covering, which is organic matrix with studded siliceous elements. And in the bottom, there is an opening of the test from which these Lobopodium come out. This is the example in Arcella. It is a brownish straw color. Then in Euglypha, this is Euglypha. It is covered by overlapping siliceous scales, the silicon material. They cover, the body is covered by these scales. And you can see these elements which are coming out over here, siliceous elements coming out from here. These siliceous elements covering, they're coming out. So this is another structure in euglypha. You can find this in euglypha. Now let us look at in foraminiferance. Foraminiferance have got reticulopodia. So the cell membranes, they extend out, extrude out like these reticular pods. They come out, and you can see that they have got an outer test covering or test. There's a cross section. This little box here, this box here, this box here. These are the tests over here, from through which the pores through which this reticulopodia come out. When it comes out, then it goes inside also. This is a very interesting point, that they come out from these holes and they go back inside the holes when it's not needed. So it's like a filament-like structure which comes out. And they form the anastomose, and they form, the anastomose means they cross each other and form a net-like structure. That is anastomose, and form a network known as reticulopodial network. So through this reticulum, you have an axis of microtubules present and forms the axial rod. They support this reticulopodia, and on this microtubule, several vesicles containing food matter or whatever is transported in both directions. Food is captured and it enters through it. It's transported through these vesicles, and therefore it has got a granular structure. Because of these vesicles, lot of vesicles are present, it gives a shape of a granule. So if you look at the test or the covering of in foraminiferance, they are organic test embedded with calcareous particles, and they have been found to be preserved as fossil records. So have a look. This is a foraminiferan. It is covered with a test covering, and through these coverings, you have lots of these anastomizing reticulopodial network coming out, basically for food capture or defense and whatever you can say. The next thing which we are studying is the axopoda in actinopodans. Actinopodans are planktonic protozoans, another type of amoeba. They have, you can see needle-like projections called axopods coming out. You have the covering, this is the test outside the membrane. And through these tests, all these needle-like things are coming out. The only difference between reticulopodia, these are needle-like and they are permanently, they come out. They don't go inside. So this is an overview of the different types of derivatives of cell membranes which are present. So with this, I end my presentation over here. Do you have any questions? Please do ask me if you have questions.
No, sir.
Is it clear, Nisab?
Hello, sir.
Ah, bolo.
Sir, general structure of a protozoan cell membrane is 6 pointer. আপনি বলছিলেন lipids, proteins, carbohydrates on the other outer surface form a coat which is extremely important in defense against
Right,
external
right.
chemical attacks. So external chemical attacks বলতে কি, sir?
If it enters inside the host system, you can have lots of acid and alkali attacking it. It forms a region known as Glycocalyx. It has got external lot of the external chemical acid alkali. তোমার পেটের মধ্যে একটা ব্যাকটেরিয়া ঢুকেছে, অ্যাসিডের এনভায়রনমেন্টে পড়ছে, তো জ্বলে পুড়ে বেরিয়ে যাবে, তো সেগুলো প্রোটেক্ট করে. So it's a covering, it's glyco. And not only that, once it enters the blood, it elicits an immune response. That is bad for that organism, but good for the host. These glycos are strong immunogens. ঠিক আছে?
Yes, yes.
Okay, then I will tell you when the next class is. Maybe in 2-3 days. On Saturday or Sunday, I will take the class. Because right now I am in Calcutta. Tomorrow I am going back to Asansol. So we will discuss it. We will talk about the next topic. Now we will try to take one from cell biology and one from this this invertebrate fast comparative analysis. we will be taking this both of them, one topic from cell biology and one topic from this paper. From in the future, we will take short classes like this. Then we can finish it very fast, and then we can talk about competitive examinations, how you can qualify in these examinations. Okay, then. Thank you. Okay. Shall I clear to
Thank
any
you, sir.
question? Okay.